So just to introduce American beech, Fagus grandifolia. This is it over here. It's a very ecologically important species in a lot of North American forests, spans many habitats ranging from northern Florida to eastern Texas, and in the north from Ontario to Nova Scotia. It's ecologically important in that it forms part of the food web. It produces beech nuts, which are a good food source for many wildlife species out there. But the ecology of forests that contain beech is changing due to beech bark disease. And you can see it affecting that tree there. You can see some of the lesions on it in that picture. One of the effects is illustrated here. It's a little darker than I would have hoped. Hopefully you can see it. Susceptible trees that are attacked by beech bark disease, one of the responses is to put out these clonal root sprouts. And so you can see these sprouts here all coming from this infected tree, and they're sort of overtaking the understory, right? And not only are they overtaking the understory, they're overtaking it with susceptible trees, right? And I guess another thing to note is that the infected mature trees are often a hazard because they're susceptible to breakage. So one of the kind of the impetus for this project is to develop a good quality reference genome, but also to develop some markers for resistance and susceptibility so that sort of woodlot managers, forest managers can make some informed decisions in terms of how they deal with beech bark disease before it comes through, but also as it's coming through, right? To be able to assess early on which trees are susceptible and resistant, right? If there needs to be some culling that needs to be done. So that's a decent introduction slide to the subject. Hopefully you all know what subject I'm talking about now. I was actually in a conversation with someone when it looked like I was going to be getting involved in this project. It's like, yeah, I'm going to be working on some beech genomics, and I had to clarify that I'm actually working on the tree rather than doing some sort of environmental metagenomics in exotic locations. Having said that, don't be sad for me, because for someone who usually spends most of the day behind a computer, we actually do get to do a fair few excursions. Not to white sand beaches, but primarily to the Arnott Forest, Cornell's Arnott Forest, which is, they're very much involved in this, providing us support and very interested in this project. So to orient you so you know where it is, I've circled Ithaca up here, got Watkins Glen over here, and Horsehead's down here. And so this is where the Arnott Forest is, roughly up against that little patch of green that is the Cliffside State Forest. And so even within the Arnott Forest, there's a map of the Arnott Forest, I can point to the exact tree that we are sequencing. So it's right on that road, that loop there. And this tree was chosen, our reference tree was chosen because, well, it's a resistant tree, and it's deemed resistant because when beech bark disease, after it sort of came through, it was one of the few trees that was left unscathed, right? So that's the one we're sampling from. There were a few others sampled, but that's the main one that we're sequencing. So I mentioned beech bark disease sweeping through, so I'll give you a little more context on that. So it's actually a sort of three-part interaction. So it begins with this beech scale insect, which is mainly Cryptococcus fagusuga. And so the beech scale attacks the trees, feeds on them. It forms these kind of characteristic white, waxy patches where the colonies are. In feeding on the trees, it essentially opens the bark up to fungi in the Neonectria genus. The fungi necrotize mainly the phloem and the cambial tissues. And these form lesions, cankers, and ultimately can girdle the trees and kill them. Right? And to give you a sense of the progression of the disease, on a stand scale, this schematic describes what's happening. So we have sort of baseline mortality pre-beech bark disease in what's known as the advanced front. The scale insect incidence starts to increase, but the mortality is still more or less what it is at the baseline. Then we have the aptly termed killing front, which is when there's high incidence of the scale insect and the fungus, and you have high mortality. And then we have the aftermath, decrease in incidence and mortality. So that's at the kind of local stand level, right? So to give you a sense of the historical geographic progression of the disease, and this refers specifically to the beech scale insect component of that. It was introduced, it's believed to have been introduced in Halifax in around 1890. It was first noted by the director of the public gardens in Halifax and spread from there, starting in 1890, reaching counties in New York State, mostly in the 60s and 70s. And now it's expanding northward a bit and also southward, right? So it's in the face of this ongoing expansion that a lot of forest managers are really interested in resources that they can use to make some informed decisions, including a nice reference genome sequence. So for that, we're using Nanopore sequencing, and we're combining the data from 4 min-ion runs, keeping reads greater than 10 kilobases. We ended up with 26 gigs of data, read length of approximately 24 kilobases, read length N50 of 24 kb, and we have our extra long read, so our longest read that we got for this was 325 kb. Just to step through briefly the pipeline or the workflow. So the assembly we're working with, do our all-VL overlapping of the reads in Minimap. Miniasm gives us the graph assembly. There's no consensus calling within Miniasm, so we use Racon for that. Medaka and Pylon then get us to the polished assembly, a few rounds of Pylon. And because we're dealing with a fairly heterozygous individual, our strategy there was to work it through purge haplotigs to get rid of the heterozygosity in that assembly. Redundance then is our scaffolding step and gap filling. And lastly, but certainly not least, we're using all maps, and for that we're inputting our scaffold assembly, but also information from a published genetic map for American beech that was published by Kalick et al. And I guess it's worth mentioning, we did also try a number of other assemblers in there. This was just the one that we ended up carrying forward, right? And here's some results. I guess things to point out here. The total length is, we were expecting about 450 megabases, 500 megabases, so the total length, we're in that ballpark. The contact N50, 800 KB, we're pretty happy with it. I'm pretty happy with it. I think it'll set us in good stead for the annotation. You can see in the BUSCOs, the benchmarking universal single copy orthologs. Our assembly is this green one over here, Fagus grandifolia, comparing it to a number of other tree genomes out there, mostly in the Fagaceae, mostly. And you can see our percentage overall complete is pretty decent. I can also point out the lower level of duplicated BUSCOs, which is evidence of purge haplotigs doing its thing. I guess in terms of the scaffold assembly, the thing that we're pretty happy about is we're expecting 12 chromosomes, and after that all map step, we were able to get 94% of the scaffold assembly onto those 12 pseudomolecules. So we're pretty happy about that. And most of that came from the all-map step. So basically mapping the markers from the genetic map onto our scaffold assembly, and then maximizing the collinearity between the scaffolds assembled in pseudomolecules and what's in the genetic map. In terms of next steps, I mean, we're still early on in the game. I need to check a bunch of those intermediate outputs, but big picture, move on to gene annotation, functional annotation, annotating TEs, doing comparisons to some of those related genomes, and ultimately the main goal, mapping target loci and finding some candidate genes, and also developing those markers that can be translated out into the field. And in terms of acknowledgments, all the people at the Arnott Forest, especially Peter and Brett, people here at BTI, Fei Wei, members of his lab, very helpful. The two Marks in the Department of Natural Resources here at Cornell, and the Arnold Arboretum and Twisted Tree Farm. And with that, take some questions before I turn it back over to Susie, I think. Yes.
Are there questions? Everyone's getting tired, feeling saturated.
Hungry, yeah.
Once you have your history on what you're going to do, you also get something for like, and like, well, where are you going to sample from? Are you going to look across that whole range?
TBD. It's a great question. Obviously, that's where we need to go. Some of that is contingent on gathering some more funding. But yeah, I think so. Another part of that answer, there are loci that are known, and there have been GWAS studies done, but they've sort of looked for the candidate genes by mapping everything onto the European beech genome, which is a fairly fragmented genome, and it's not the American beech. So one of the things we can do is just figure out where and what those are within the American beech. That's a sort of low-hanging thing that we can do to get started. And then, yeah, we'll have to presumably do some broad sampling across the range and resistant and susceptible plants. And yeah.
Any other questions? Let's thank Adrian and all our speakers again.